Hi class, this is your chapter 5 lecture presentation. Chapter 5 is on the cell membrane, so membrane dynamics, talking a lot about membrane transport. We are also introduced to the concept of osmolarity here, which you learned about in lab exercise 3. So this content will be helpful for that lab exercise that we do where we're studying the concentration of salts, the effects of those salts on living cells. We will go through pretty much the entire chapter, but as always, please focus on the material that I include here in your lecture presentations, your lecture slides. The figures that I put in here are the ones that I really want you to focus on. The book often has plenty more, but I choose these ones because they are have the information that I need to teach you. Okay, so we've learned about homeostasis, and we know that it's a dynamic process, but we know that it doesn't mean equal, right? Equal would mean 10 units inside, 10 units outside. It does not mean equal at all. So let me show you on the board here why that is. So here is an example of one of our cells, so a human cell. So we've got our intracellular compartment, the intracellular fluid, and then we have the extracellular fluid outside the cell. So for osmotic equilibrium to occur, let me show you, give you a kind of a cartoon drawing of what that means. So in order for our cells to be in osmotic equilibrium, the majority of the potassium is inside the cells, where there's just a little tiny bit of it outside the cell. So the big letter means there's more of it. For sodium, however, it's the opposite. There's way more sodium outside the cell than there is inside the cell. There's just a little bit inside. Same with chloride. It's present mostly outside, just a little bit inside. The other thing that's inside the cell and is present in much higher quantity are proteins. And then there's a little bit of them out here. So this is a picture of the relative concentration of some of the major ions that are important in physiology. The majority of the potassium is inside the cell, and the majority of the protein is intracellular, but the other ions are present in higher concentration in the extracellular fluid. That is what is necessary to reach osmotic equilibrium. However, though, note that there's no chemical equilibrium here. In fact, the chemical concentrations are very different, right? Because we've got the majority of potassium inside and just a little outside. So in order to achieve chemical equilibrium, the cell has to be in chemical disequilibrium. And it also needs to be an electrical disequilibrium. We'll get to that a little bit later in the lecture. So remember that equal would mean the same inside and outside. So 10 units of potassium in, 10 units of potassium out. And that's not at all how it is. In fact, our body won't function right if we don't get back to this. All right, let me pick up the slides again for you. So the balance between the intracellular and extracellular fluid, again, in order to achieve osmotic equilibrium, oh, I did spell that wrong. Sorry about that. The cell must be in, and in chemical disequilibrium with the extracellular environment, as well as in electrical disequilibrium. So we'll come back to the part about the electrical disequilibrium in a moment. It ties directly to the ion concentrations, but I'll show you that later on in the lecture. So let's look at a diagram here of the fluid compartments of the body. So we've got our intracellular fluid, which is everything inside the cell membrane. And actually the majority of the fluid is here. 2/3 of our fluid volume is within the intracellular compartment. The other third is in the extracellular fluid, which includes the interstitial fluid. See, that's that kind of blue purpley fluid that every single cell and tissue is bathed in. That's the interstitial fluid. It's like a buffer to our cells. And then also we've got another extracellular compartment, the blood plasma, which is contained in the blood vessel. There is leakiness of the blood vessel, and there are different conditions where our blood vessels become more or less leaky. But we need this to happen in a controlled manner so that, for example, the red blood cell may leave the blood vessel and go through the interstitial fluid to the cell so that it can deliver oxygen to it. So we do see a normal amount of leakiness in our blood vessels. So our body is at osmotic equilibrium in these 3 fluid compartments. All right. So you know that water, we're made majority out of water, and osmosis is that passive movement of water along the gradient of solute. And it can't happen by itself. It does go through the membrane very, very, very slowly, but that just isn't fast enough to support life. So instead it uses a process of facilitated diffusion. So it has its own protein, aquaporin, that it follows through. So our body has a number of different mechanisms to control water balance, and we'll learn about this all semester long. There are actually differences in the regulation of water and balance of water between gender, between in different ages. For example, you've probably heard that the elderly are more prone to dehydration. Well, that's because one of the reasons why is because protein synthesis slows down. So we don't have as much protein being produced. Muscle mass is lost, so that amount of protein is lost too. So that affects the osmotic regulation and distribution throughout the body. So because our body and our cells are majority water, that does provide the right chemical environment to help moderate some of the changes that we would see. I mean, again, think about the example of living near the ocean, where the temperature changes near the ocean aren't nearly as dramatic as what happens in the desert. And the reason why is because there's a lot of water vapor present near the ocean. So water is able to moderate temperature very, very well as other things, not just temperature. That's just one example. So tonicity is a term that we use to describe the relative concentration of solute in the environment. Tonicity always describes the environment. It is incorrect to use the word tonicity to describe what's happening inside the cell. We have a different term for that. And tonicity ties very importantly to what are called non-penetrating solutes. And these are materials that, just as the name suggests, they're not able to get through the cell membrane, at least on their own. So we'll go through the list of what falls into this group a little bit later. But since we are talking about tonicity, it's appropriate to teach you at this point that it is tied very, very well, dependent upon the concentration of those penetrating solutes, things that can't pass the membrane. So looking at the distribution of the fluid in our body, probably the right figure here is a little bit easier to see. So 2/3 of the fluid is intracellular fluid, and then the other third of the fluid is extracellular fluid, with about 75% of the extracellular fluid being interstitial fluid and only 25% being plasma. Okay, so now let's get into that discussion about osmolarity. So we know osmosis involves the passive transfer of water, but let's tie it to osmotic pressure. There is a pressure associated with the concentration of solutes and movement of water. So this is basically a syringe where we can take the plunger out on one side to increase the volume. There is a selectively permeable membrane present, and then we've got glucose in a watery solution. And you can see there is much more glucose present in solution B or compartment B than there is in compartment A. And this is how we're starting. But as water moves by the process of osmosis, right, that's going to happen. Now what happens is we've lost some of our fluid in compartment A and gained it in compartment B. But that has we've increased the volume here, right? So the volume has increased to allow for that increased water. So the pressure dynamics don't change. But what happens if the volume can't increase, right? What if we don't have a mechanism to allow the cell to increase that much? Or what happens if we have a force that goes against it? So here in the bottom panel, we're looking at a force that is being applied in the opposite direction of osmosis that is equal here to the pressure. Let me say that again. It's a force strong enough to oppose osmosis. So that is the osmotic pressure. And you can see in this example that now all the solute, all the glucose has gone into compartment B, but we still have a good amount of water in compartment B, right? It's different from how we saw it in the starting position. So what we're seeing here is that this osmotic pressure sometimes is strong enough to actually draw water into this hypertonic environment and to try to dilute it out. So we call that opposing pressure is called hydro for water, and static because it opposes it. That is called hydrostatic pressure. And right, this is obviously a model, but this does happen with our cell membranes. All right, so we know what molarity is. It's a concentration, moles per liter. What osmolarity is, it's very similar. It's a concentration as well, but it's the number of osmotically active particles. And sometimes that we need to consider the environment and what type of solute we're talking about. So as we progress through the course of this chapter, we'll look at some more complex examples of that. So, but I'll keep it kind of simple to begin with. Some textbooks you'll see it referred to as osmolality. It's essentially the same thing. It's really, the two terms are used interchangeably, but just in case you see it as osmolality in your textbook, please know it's being used in place of osmolarity. We will use osmolarity. All right, so I'm gonna pause the recording so I can erase here. And I wanna just draw a picture to review tonicity real quick. So give me a quick moment, I'll be right back. Okay, now we're looking at tonicity. So tonicity pertains to the solution, to the environment, every time. If you need to memorize that, please do. we've got our three prefixes, hypo, iso, and hyper. We'll actually begin with iso. So iso means the same. So what we're talking about is the same concentration of solute or osmolarity in the extracellular compartment as there is in the intracellular. I'm just going to make up some numbers so we can have a quantity. So say we have 100 units inside the cell. That means we also have to be isotonic 100 units outside the cell, because the tonicity describes the environment. So there's no net movement of water, right? The amount going across the membrane in the cell is equal to the amount going out. So the cell stays the same size. So there's no change in cell size. In the hypo environment, so in this case, say, let's put some numbers to it. We've got 10 units outside. Hypotonic means less outside. And then we've got 100 units inside. So what's going to happen is that the water from the environment is going to go inside the cell to effectively try to dilute the concentration inside the cell. And the cell will get bigger and bigger and bigger and heavier. We measure that in lab exercise 3, and eventually it will burst. So it gets bigger, and then it undergoes lysis. So it gains water. And then in the example of the hypertonic environment, so now we've got more outside than there is inside. So the cell is essentially going to try to dilute the environment. So water is going to run outside the cell. And sometimes that's enough. And what happens here is it shrinks. And it eventually will shrink so much so that it, the membrane falls up on itself and it falls into pieces. So both the hypotonic and hypertonic environments are capable of inducing cell lysis, but through different mechanisms. In the hypotonic environment, the cell literally blows up and bursts, it pops. Here it falls back on top of itself and then just falls apart. So that's tonicity. Now, I recommend you have a solid understanding of tonicity before we move to osmolarity. That's why we're doing it first here before going to osmolarity. Okay, let me get everything away. Oops. So just as we reviewed with tonicity, we can apply these same prefixes to osmolarity, and they do mean the same thing. Hypo always means less than, iso is the same, and hyper is more than. I will go through an example once we have a little bit more knowledge under our belt. So in order to determine osmolarity, we need to know a couple things. We can't just look at it so quickly. So one of these things we need to know is, does the molecule dissociate? Does it fall apart into more than one molecule, one particle? For example, sodium chloride, a salt held with that ionic bond, if you put it in water, it will fall apart into sodium and chloride. You've created two particles. We'll look at examples, don't worry. So you need to know if it's going to dissociate or if it's going to remain as one. I mean, really, you're saying the same thing, but you need to know if that particle is going to fall apart in a watery environment or not. Okay, so now let's look at the example of some of the penetrating and non-penetrating solutes in our 3 fluid compartments. It should be no surprise. Hopefully you're already thinking, hey, they should be different because Dr. Mitchum just told me that there's more potassium inside the cell than there is outside. So you already know there's a difference for sodium and potassium and chloride. So I put a little list here. Let me explain it because there's 2 caveats on this list. So examples of penetrating solutes include lipids, not giant, huge ones, but most lipids can go through. Most nonpolar solutes can go through. So molecular oxygen can go through, which is nonpolar because it's symmetric. Our respiratory gases can go through. Like methane is an example of another solute that can go through. Urea goes through. Water can go through. But the reason I italicize water is because it's not a solute, but right, we need to always keep in mind that osmosis is going to happen. Oops, I'm so sorry. Examples of non-penetrating solutes include anything with a charge or anything that's big. So all of the ions are going to get stuck. Proteins can't go, they're too big. ATP is going to be needed. ATP is too big itself, but it's also going to be needed to move things against the membrane if they can't follow through using diffusion. And then glucose is here, and glucose is the second caveat. Glucose is technically a penetrating solute. It will penetrate. However, because of its size and chemical characteristics, it penetrates so slowly that it's not relevant for what's happening at that time with the other solutes at the membrane. So effectively, it behaves as a non-penetrating solute because it takes so long, but it is correctly a penetrating solute. But when I show you how to work problems, I always put it in the non-penetrating column because everything else is going to go so much faster. So you have 2 completely different time frames. Okay, so looking intracellularly, yep, there's potassium, purple. It's in high concentration, but look how low it is in the 2 extracellular compartments. And then our other ions are opposite. So sodium, chloride, and then this is bicarb. They're all low intracellularly and then higher outside. And proteins are higher inside than they are outside. Now, our plasma has a good concentration of plasma proteins in it. So we do see some there, but they essentially, that's where they go. They are plasma proteins. Okay, so this is a table out of your textbook, and essentially we need to memorize it. These are the rules when you are looking at the relationship between osmolarity and tonicity. You may not predict osmolarity from tonicity or vice versa all the time. We get one case where we can do that. So we need to understand what to do in the rest of the situation. So we begin with the assumption that everything inside is staying there. So you assume that everything inside the membrane is non-penetrating. It's staying in there. For example, a protein is staying in the membrane. Now, osmolarity is going to happen. So that means that osmolarity is determined before you consider the effects of the watery solution. Okay, let me say that again. Osmolarity is determined prior to putting the solute in the solution. That means it can't break apart yet. Okay, that's really important. Osmolarity is assessed prior to being put in that watery environment because it will achieve osmosis, right? So what would be the point of evaluating it after? Because it's going to be in osmotic equilibrium. So we know what tonicity does. We just reviewed that. It describes the changes in the cell volume with regard to the concentration of solute in the environment. Tonicity is always environment. So to apply the relationship between osmolarity and tonicity, what we need to do is we're looking again at the tonicity of those non-penetrating solutes, yes, inside the cell, but in the solution. right? If something can get trapped inside the cell, something could also get trapped outside the cell. So we know that the movement of water is going to go to the higher solute, to the higher amount of non-penetrating solutes. The one time where we can correctly predict osmolarity and tonicity is in the hypo situation. So a hypoosmotic solution will always be hypotonic. So those always hold true. But everything else, you've got to look at your concentrations of solute prior to osmosis, and then you look at it after. So essentially, the rule of thumb here is osmolarity is assessed prior to exposure to water, where tonicity is after, right? Because tonicity is the change in cell volume due to the relative distribution or concentration of non-penetrating solutes. I know that's a lot. I encourage you to write this out, replay this part of the lecture, but really the best thing to do is apply it to problems. So you do have a homework assignment where you are asked to actually draw a solution and represent the different concentration of solutes in different conditions. So hopefully that will help you with this. Okay, so let's look at an example of osmolarity. So we've got three solutions. We've got solution A, B, and C. Solution A is 1 osmolar glucose. Solution B is 2 osmolar glucose. And then C is 1 osmolar sodium chloride. So now let's describe some of these relationships. And there is more than one correct answer for some of these boxes. So A is hypoosmotic to B because 1 is less than 2. So hypoosmotic to B. We could also say that B is hyperosmotic to A, right? That's the same thing. Now A, 1 osmolar glucose, and C, 1 osmolar sodium chloride, that's the same. So they are isoosmotic. Remember, we always look at osmolarity prior to the cell or the solute being exposed to water. So that means something like sodium chloride has not had time to disassociate yet because it's not in that environment. So therefore, A is iso to B. We could say that C is hypoosmotic to B. That would be another correct relationship. So that's what you can kind of run through all of these. I would encourage you to actually draw them and keep it simple, but you know, one solute or 2 solutes. Okay, so moving forward. So now looking at the tonicity, this is a little bit easier. So we've got several situations here. So we've got solution A, B, and C. I copied the information over from the previous slide down here. But you don't even need that. You're told that the cell swells when it's put in solution A. So what cell swells? The hypotonic solution is a cell put into a hypotonic solution swells. So you don't need any of that information. In fact, it doesn't even help you because you don't know anything about the the concentrations of the different solutes yet. But just knowing that the cell swells when it's put in a solution tells you it's going into a hypotonic solution. If it goes into a solution and it doesn't change, well, you know you're in your isotonic environment. And if it shrinks, now we're going into that hypertonic solution where the cell is trying to give off its water to dilute the environment. So it's hypertonic. So often tonicity is a bit easier to understand and a bit more straightforward for students, but it becomes complex when we start to look at our disassociating solutes, things like salts. Okay, so we've got a number of transport processes, right? We're talking about the movement of our different ions across the cell membrane, but how do they actually get there? We've just learned that ions are non-penetrating because they are charged. They're just going to get stuck in the cell membrane. So we've got a number of different processes, transport processes, and sometimes the same material has more than one process it can move by. So bulk flow applies to fluid and gases. And it just describes that diffusion following a gradient, right? Anytime we're talking about diffusion, it's a gradient, but it's not just a solute gradient. As we've learned, osmosis also has a pressure associated with it. So it's a pressure gradient. So the respiratory system uses bulk flow. So I'll point this out throughout the semester. But you know that the cell membrane is selectively permeable. It lets some things through, but not others. So we have passive transport, which is diffusion, right? Simple diffusion. But then we've got our protein-mediated diffusion too. We've got our facilitated diffusion, like water does. We've got examples of ion channels that allow ions to flow through passively. Here's the name of the one, facilitated diffusion for water, aquaporin. So that's how the majority of our water moves. But we've also got our active processes. So we've got the movement of things in vesicles, and that uses a lot of energy. So we've got exocytosis, endo, and phago. We'll look at examples of all of these. But even we've got our transporters, but we can have direct as well as indirect or secondary transport. And the basic difference with direct or primary transport is it uses ATP directly. So it's breaking ATP down. Indirect or secondary transport, it uses a gradient that's generated by ATP. So it's, I'll show you an example when we get to a good one. So it still is using ATP because it can't happen without it, but that process has already happened, and then there's a gradient built up, kind of like what happens with the movement of hydrogen ions through ATP synthase, as we'll see. All right, so diffusion is passive. It follows that high to low gradient. We've talked about the role of different chemicals and how a lot of times we're in chemical disequilibrium to achieve osmotic equilibrium. So what are some of the factors that affect the rate of diffusion? We saw this in the first beginning of I think it was lab 2. So things like distance definitely do. Temperature does. When it's warmer, things move around faster. So usually temperature increases diffusion, as long as we don't denature and break apart the molecules we're talking about. And then molecular weight, the size of it is a big factor. Things that are heavy move slower. And then the membrane itself. I mean, there's different characteristics of the membrane that influence the rate of diffusion. So we've got a number of factors. So ions can't move by diffusion because they are charged. But they are tied so importantly to the electrical gradient. They create the electrical gradient. And an ion, you know, opposites attract, positive ions to a negative ion. And that's true for charges too, positive charge to negative charge. You can think about the example of two magnets. If you put the positive to positive end, they repel. You have to put positive to negative. So the movement of ions does use both, or it really creates both the chemical as well as the electrical gradient. So we can call that the electrochemical gradient. They're hard to separate out, and you'll understand why. You'll really understand why at the end of this course, but hopefully you'll have a better understanding of this too after we get through this chapter. We'll apply it to a real system when we move into chapter 6. So we'll study it kind of more in the human body as opposed to, you know, the nuts and bolts pieces of it. All right, so let's just remind ourselves of the cell membrane. It's that phospholipid bilayer. So we've got two polar hydrophilic regions where the heads are, and then the hydrophobic tails are inside. So if we're talking about simple diffusion, that would just be a gradient across the membrane. But size is a factor. So something that is small can go through where big things often just don't even get in. Solubility is a big factor. So our fat soluble materials are able to get through much easier than water soluble materials, because water soluble materials are going to move into this region and then just get stuck, and eventually the membrane would disrupt. The surface area is also a factor. If there's more area, then diffusion happens faster than if all the solids have to funnel in to the same area. So we've got a number of factors here. We've got the solubility, the molecular size, the gradient in itself. The steeper the gradient, the faster diffusion. That's always true. The surface area, and then the composition of the lipids. So I would like you to more focus on this part of the slide rather than this. We can use Fick's law and this calculation for membrane permeability. Honestly, what I would rather you focus on is understanding the structure and function relationship of what's happening here. And diffusion is an excellent example of studying an excellent functional example to study the structure of the cell membrane. All right, now proteins aren't going to go by diffusion because they're just too big. So they are happening through, well, some of them can go through facilitated diffusion, but most of them are going to go through active transport. We do have some examples of proteins, smaller ones that go through, facilitated diffusion. So let's look at some examples. So if we take all of our membrane proteins, all of them, and group them together, we have different functions, right? We've got some that form transport functions. We have some that are structural, some that are have enzymes in them, some that have receptors in them. I'm not going to walk through this map here. I don't really think it lends itself well to me just pointing and saying the words. But what I would like you to use this for is after you've studied, after you've completed your activities for this chapter, come back to these membrane maps or these different maps, and then try to understand an example of a carrier protein, or the example of a receptor that does receptor mediated endocytosis. Even if it's a general receptor, just with the structure of a Y would work. So I think that these have value as a way to kind of come back and test your concepts when you're looking at how well you've learned this material. Okay, so here's an example of receptor mediated endocytosis here. So it involves a vesicle. So first of all, what happens, we actually begin here. So now the receptor binds its matched receptor, and this is by shape. These are usually majority proteins. Sometimes there's sugar groups present, but the majority of the molecule is usually protein. So it's all about shape. So the binding of the ligand to the membrane will often send other signals to the cell to do other things, but it induces the process of endocytosis here. So you can see that the membrane actually bubbles in and then pinches off. So the physical length of the membrane has gotten shorter because some of it is right here inside this vesicle that contains the ligand that's still bound to the receptor. In most cases, our body is really smart and efficient. It takes a lot of time to make and fold proteins. So it's going to save this receptor and shuttle it back up so it can be used again most times, unless it's damaged. So this is going to use energy. We're not seeing it here, but this is a energy dependent process, endocytosis. All right, so water, we talked about our water channel. That's an example of a channel, aquaporin. We also have channels for ions, and we have different ones. We have some ion channels that allow one thing, some allow two things. We'll learn about the different examples as we move through. So what we're doing is building a list of the different types of passageways that are in the cell membrane where water is associated, where water moves through. So we have what are called open channels, and this would include leak channels and then pores. We'll look at examples. We have gated channels. So open, there's no gate. A gated channel, there's a gate. So you have to get by the gate. So we have chemically gated channels, voltage, and mechanically gated channels. And we'll look at examples of them. All right. So looking at a channel protein. So this is just a generic example. So we've got our extracellular fluid in the light purple, the cell membrane is the darker purple, and then the yellow is the interstitial fluid. And this is an example of a channel protein that allows water to move through. The example of an open channel is on the bottom right. So there's no gate. The fluid can just move through. But a gated channel, we've got an open position where water and whatever's in it can move through, or a closed position, right? And we've got different conditions that can close and open this gate. Now, let's look over here. I think this is actually a little bit easier. So we're looking down, and this is an example of aquaporin. So we've got the protein that interacts with water, or whatever protein you're talking about, lines the inside of it. And then you can see the glycerol heads and even the lipid tails, but behind them. So the importance of including this figure is to show you that these channel proteins span the entire membrane. So they connect that extracellular to intracellular environment, and whatever is passaging through them is shielded completely from the rest of the membrane. So that means the chemistry that is present in the tunnel is determined by the proteins that line or make up that tunnel. All right. Right. If water is moving through it, though, those proteins need to be hydrophilic, right? Because otherwise water's not going to move through. So that's an important thing to consider. In this example, the channel is made up of all the same type of protein because it's the same shape and color here. But other channel proteins can be made of more than one chain. All right. So now looking at examples of carriers, those were channels. Now we're going to look at carriers. We have three categories, and we'll look at examples. But these allow for the movement of something through its gradient. So we're still talking about diffusion. An example in our body are the GLUT transporters, which we'll learn about. Okay, so looking at a generic example of a carrier. So these work kind of like a clamshell where it's open to one side and then the solute gets in and binds, and that changes the conformational shape, and then it opens to the other side, as you can see in this top generic cartoon of a carrier channel. So notice the proteins of the channel do completely span the membrane, allowing whatever moves through to not touch the membrane. So a uniport carrier is represented by this glucose transporter here, and it means everything moves in one direction. So only one thing moves through in one direction. Symport can move two or more things, but they're going in the same direction. So we've got a sodium glucose transporter that allows sodium and glucose to move from the extracellular environment to the intracellular environment, in this case by diffusion. And then we've got the example of an antiport carrier, meaning that they're going opposite directions. So we're moving two different things opposite directions. And this is exemplified by the sodium-potassium pump And this does require ATP, so this is not diffusion, it's active transport. So those are carrier proteins. I'm gonna flip us back a minute. Sorry. Oh, I didn't realize it was this far. So going back to this figure, we've now reviewed carrier proteins and then channel proteins. So you'll be able to fill in this content with your knowledge as we get through it all. Sorry. All right, I should have moved here because here it is bigger. So this is the uniport moving one thing in one direction. Symport, sim meaning together, and it's moving two things in the same direction. And then the antiport co-transporter moves two things in opposite direction. So both of these guys are considered co-transporters because they're moving two different things. So we can think of our carrier proteins, just to give you another example to relate them to, kind of like a gate. So this could be the Panama Canal. So we have in the top panel, we've got our ship that's going through. The first gate opens and it allows it to move into the channel. So now it's in there. But when the second gate opens, it controls the direction of passage, right? So the direction of passage is to the other side of the gate because of how the two gates open. I mean, if we're in this middle situation where the boat is trapped with both gates closed and only the first gate opens, the boat's gonna have to turn around and go that way. So the carrier protein controls the direction. And we saw that with the symport and the uniport. So active transport can use a carrier protein as well. And we're talking about going against the gradient since we're using ATP. We see a conformational change here too in the protein, the carrier protein, but now we need energy, and it can either be primary or secondary transport, but either way it requires an ATP molecule. So primary or direct active transport utilizes the ATP molecule directly itself, meaning that the ATP needs to be there. And then in secondary or indirect active transport, now what's happening is the gradient that was built because of ATP is being used to facilitate the movement of a molecule. So it's not breaking down the ATP directly, but it couldn't happen without the role of ATP. It's just next in line in a sequence. So some examples of active transporters. Here's the one we just spoke about, and we're going to learn more about it as we continue through. We'll actually talk about it all semester because all of our cells have it. This is the sodium potassium pump. So when you see something that uses ATP, the word pump is synonymous. So if I just say sodium potassium pump, you know that that is an ATP dependent transporter. We've got a calcium pump, a proton pump, and then a proton potassium pump. And so we've got examples of different directions. So these are all examples of primary active transporters that directly use ATP, meaning it needs to be there. So we'll look at an example utilizing the sodium potassium pump. So it is present in the cell membrane. It's got way more than one copy, but we're just keeping it simple. So inside the cell, there is a lot of potassium and only a little bit of sodium. And the opposite is true outside the cell. This goes to what I drew on the board the first time. So outside the cell, we have way more sodium, these little yellow circles, and just a little bit of potassium. So the pump is moving each of these against their gradients. So we are putting potassium into the higher concentrated place, and we are putting sodium into the higher concentrated place. So that's why these are both required, these utilize ATP because we are going against the gradient. and they are considered primary or direct active transport because this transporter itself breaks ATP down into ADP, adenine diphosphate, plus that phosphate, so that it can use it to do this. All right, so now let's dig a little bit deeper into the sodium-potassium pump. It actually exchanges a different amount of potassium than it does sodium. So it moves 2 potassium in exchange for 3 sodium. So it's not 1-to-1. And this is actually really important in establishing the osmotic equilibrium that's necessary for our cells to function. So let's begin here. And oh, my mouse is lost. Sorry, I'll get it back, don't worry. But I gotta clear you. There. Ah! Sorry about that. Apparently I stopped the screen share. Okay, back to that. So what happens, again, we're going against the gradient. So sodium is moving outside to the extracellular environment where it is highly concentrated. So 3 sodiums move into this pump and they have binding spots. So once that happens, a conformational change is induced by breaking ATP down into ADP plus that phosphate group. So here's that phosphate group. It is what's needed to cause the conformational change. And then the sodium are able to move out, and in come the 2 potassium. And then we go back to the beginning. So it's literally changes shape. But it does require the breakdown of adenine triphosphate to adenine diphosphate so that the inorganic phosphate group may be used to power this this reaction. So the phosphate actually induces the conformational change of the gate to open from one side to the other. Now there is different affinity for the binding between for even the same ion. We sometimes see binding sites that have high affinity and sometimes binding sites that have low affinity. We'll see more and talk more about that as we move into our muscle section. So I'll just defer to on that topic until we really move through. What we need to understand here is that the binding is strong enough to hold the solid in its place, but not too strong so that it can be released and moved across the membrane. If it was so high affinity, it would never come off. Okay, let's look at another example. So this is the sodium glucose cotransporter. So it is an example of a secondary or indirect cotransport because it is not using ATP directly. Instead, it's using a concentration gradient that was built up from ATP in a previous step. So, so we're looking at sodium. So sodium goes in and binds inside. It is moving it into the intracellular environment. So sodium binds. And that when sodium binds, notice that there's no binding site for the glucose yet. But when sodium binds, now we get a binding site for glucose. So glucose comes in and binds, and that induces the conformational change. And now our cotransporter is open to the intracellular environment where the sodium and the glucose are released. So there's no breakdown here of ATP anywhere. It's happening utilizing energy that was built up from this sodium gradient that's already there. So we've got a high concentration of sodium inside the cell that's outside the cell now that's driving again, right? That drives the movement. Sodium's higher outside the cell, so that's going to drive the movement inside the cell. So there's the gradient that causes that to happen. So it's that gradient of sodium that that follows through the process of diffusion that allows the movement of the glucose. So really it's just the glucose that's going against the gradient in this example. That's an important distinction to make. It's only glucose that's going against. So your book gives you the relative concentration of each. The brackets always mean concentration. So it's telling you that the concentration of sodium inside this kidney is high, where the concentration of glucose is low. Just in case you need a little help interpreting how to read those figures. Took me a little bit to figure them out. So I'd like you to think about this as you go through. So how does something like specificity and competition and saturation play a role? Because these are all terms that are relevant to protein binding, and these transporters and carriers and channels are mostly protein. So they do apply here. There's actually a question in your homework 2 assignment on membrane dynamics and osmosis that, or osmolarity that pertains to this. So you're in the part of the textbook that, that will help you to answer that. All right. We're already seeing examples of specificity in that the sodium binds in the sodium site and the potassium binds in the potassium site. So let's look at an example of competition. So here we have glucose and maltose, right? Maltose is made of 2 glucoses. So glucose can bind here, but so can maltose. And if maltose binds, look what it does. It completely blocks glucose from binding. So we do see examples of competition where similar molecules can bind the same transporter, and whichever one gets there first, which is usually whichever one is in a higher concentration, is going to facilitate that. So it's only when glucose is present does it actually transport, but maltose is inhibiting it. So maltose is considered a competitive inhibitor. It's competing for the binding site. So an example of saturation is seen here in this graph. So on the x-axis, we're looking at the substrate, the extracellular substrate concentration, so what's being moved through the transporter, versus on the y-axis, we're looking at the rate. So as we have more of the substrate, it's able to move because of the gradient. Let's go back. So if we have a bunch more glucose out here, it's going to move quicker because it's going to get to its binding site quicker because the gradient is steeper, and that influences the rate of diffusion. This will happen though until we hit maximum. Once all the binding sites are full, no more can have an effect. So it doesn't matter how much more is there. If the transporters are full, if it can't find a seat on on the bus, it can't ride the bus. So our transport rate is proportional to the concentration of substrate at saturation. We'll see some better examples of this throughout the semester. All right, so the process of phagocytosis, that's big, uses vesicles, and those are made out of materials from the cytoskeleton. So this is one way that bacteria and other material can be engulfed in by our cells. So let's look at this process. So here we've got a phagocyte, so a cell that's capable of doing phagocytosis. This would be one of our immune system cells. Inside it has the lysosome, which contains digestive enzymes. So here's a bacteria, and it binds onto the surface of this cell, and you can see the membrane is starting to invaginate in, it's pinching in, and the cytoskeleton of the cell is at play in doing this. Eventually it buds off, forming the vesicle. So some of the membrane is lost and is now inside the cell surrounding the bacterium in this vesicle. Eventually the vesicle is going to fuse with the lysosomes, and that allows the spilling of the digestive enzymes that are in the lysosomes to go into now the phagolysosome because they're fused, and that kills the bacterium. It breaks it down into small enough pieces which then can be released to the extracellular environment. So yes, this is an ATP dependent process. It's a very expensive process in terms of energy, but it's also a very effective process. We get a lot out of this. Not only do we get rid of the bacterium, but this is one of the major mechanisms that our immune system uses to generate its immune responses. So that way, hopefully, if a person's infected with the same bacteria again, they won't succumb to becoming sick. So endocytosis is just a smaller, same thing essentially, but on a smaller scale. So it forms phagosomes, or excuse me, it forms vesicles, but they're much, much smaller than what we saw. So penocytosis is an even smaller process of it. Another form utilizes the receptor, and that would be receptor-mediated endocytosis. There's a protein that's present, clathrin, and it is what interacts, and I'll show you. It's easier to show you. All right, so we're going to get to it in just a minute. So exocytosis is the opposite, moving to the exit of the cell. So it's used to get rid of big molecules and waste material that's in those lysosomes. And it's pretty much always happening. So it's constitutive. Cells are always doing it. Okay, so let's look at all of that together in this one big figure. So we're going to begin step one. So our yellow ligand binds to its matched receptor, and that will send signals inside the cell, but it does induce the invagination of which will lead to the endocytosis. So here's that protein I just mentioned, clathrin. It is present inside the cell surface at the area where the receptor is bound to its ligand. Sorry. So that is the signal to the cell to know where to pinch off and create the vesicle. So now we've got an example here where there's two receptors with their ligand inside the vesicle. The clathrin is going to go back to the cell surface so it can be used again. Anytime our body can reuse a protein, it's going to do it because proteins take a lot of time and energy and materials to make. So it's gone back. And now our receptors and our ligands separate from one another. And look, our receptors go back. So they're being transported back to the cell membrane through the process of exocytosis. So this is endocytosis bringing in. The cell membrane actually gets smaller because it pinches off to surround and create the vesicle. But then it gets bigger on the side of the exocytosis because it bonds back in, expanding that cell membrane. So that's the process of both endocytosis and exocytosis. But we see here two examples where proteins are recycled. And again, that's pretty typical, providing the protein is not damaged. So the ligands are going to go to the lysosome where they're digested down, or sometimes they're even sent to the Golgi apparatus, where they can be sent to other parts of the cell if they're needed elsewhere. Okay, so applying this to a surface, an epithelial surface, there's definitely a direction in our epithelial. So they're polarized, right? That's what polarized means. One way we can view it. So we're looking here at the side that is exposed to the inside of the intestine or kidney. So this is inside an organ. And then we've got our epithelial barrier. We can see that we've got wiggles in the cell surface membrane to increase the surface area to volume ratio. So that means a lot more material can move through, because if we have more surface area, then transport's better. And then we've got, of course, got our nucleus, and then we've got our extracellular fluid. So what this is showing us is that different material moves by different processes. So we've got some examples of transport proteins moving here. So this is a transport protein. This is a transport protein. So they're very specific. They're only letting the green circle through. We've got our tight junctions that build this tissue. We've got our tight basement membrane that is that barrier between the epithelial and the extracellular fluid. We've got examples of, so we've got another transport protein here, different shape. It lets through the red one. And then we've got examples of what is called, in this arrow here, this is an example of absorption, moving out of the intestine. So we eat, we absorb our nutrients out of the intestine into the bloodstream. Paracellular is it's going across on one side and it's open on another. This is also absorption. Transcellular, it's going through two membranes, so it's transversing both sides of the membrane. And then the opposite is secretion. Secretion is when we take material out of our body and put it in the path to be eliminated to the environment, either the intestine or the kidney. So we've got a number of different transport apparatuses present on our epithelial surfaces to allow for the movement of different materials across them. So at times we're utilizing diffusion, at times there's passive transport, and then that could be either primary or secondary. It really depends upon what molecule we're talking about. So think about the questions that I put in here. So why would it might be necessary to use that secondary active transport to move materials across the surface when getting glucose out of the intestine and putting it, moving it to the intercellular environment? Well, what happens if we don't have a glucose gradient? Well, then you can't do diffusion. So secondary active transport is a way that, that glucose could move dependent upon, say, the sodium gradient that's created through that sodium glucose transporter. So penetrating solutes are able to diffuse across the membrane until they hit their equilibrium. So that would include, again, the lipids. Most non-polar solutes are respiratory gases. And then I put water here, even though it's not a solute. Our non-penetratings would include our proteins, things that are charged. So our ions, our polar molecules, things that are big, including big fats. And then glucose, since it moves so slowly, it really behaves as a non-penetrating solute. So when you're working problems doing osmolarity and tonicity, you want to consider glucose as a non-penetrating solute, unless you're told that the experiment went for several hours and allowed the time for glucose to transport. All right, so now that's a lot of information. Now we need to actually apply it to a situation. So this takes us back to where we began. So I began the lecture on the whiteboard. We looked at a cell and we looked at the intracellular environment and the extracellular environment. Let me just pause so I can get the board set up again. Give me a quick moment. I'll be right back. All right, so getting back to our, learning about the electrical potential or the resting membrane potential of our cells. It is established in part due to the concentrations of these ions, also due to the presence of proteins in the cell. So the big P with a minus represents protein concentration, and most proteins have a negative charge. So the presence of the majority of the proteins, the negative proteins in the cell membrane, has a lot to do with the electrochemical charge. But the other ions, the ions are important. What also is important is that sodium-potassium pump we've been talking about. So what happens is we end up with a negative charge building up inside the cell membrane. And it literally aggregates at the inside of the cell membrane. And it's due to it has a lot to do with the presence of those negative proteins. But what that does is it attracts the positive ions to it. So we end up with a positive environment right outside the membrane. And as you know, opposite charges are attracted. So this is the electrochemical gradient that is established due to the distribution of the different ions and those negative anionic proteins in their different compartments where they usually reside at homeostasis. We need to also consider the role of that sodium-potassium pump, though, because it's present. And the sodium-potassium pump is going to move sodium and potassium against their gradient. So it moves potassium in and sodium out. But keep in mind, it's not 1-to-1. It's 2 potassium for every 3 sodium. So we're taking more positive out. Taking positive out, what does that do inside? Negative. So all these things tie together in creating this resting membrane potential. And our different cells have a slightly different number associated with it. So our nervous system cells have 1 resting membrane potential, but our muscle cells have another, and our cardiac muscle cells have still yet another. All right, let's get back to the slides. So overall, you know, our body is electrically neutral, but we have to have this electrochemical gradient. And it comes because of that disequilibrium of chemicals. Again, different ion concentrations, different protein concentrations in between the intracellular and extracellular environment. But also the potential energy that can be harnessed from that electrochemical gradient is important. So the resting is kind of the homeostasis. It's the baseline. And so we'll start at homeostasis, our resting membrane potential. And if we plot over time, we'll see changes in the voltage up and down. Now, the most important ion we say in establishing the resting membrane potential is potassium. And it can sometimes be confusing because potassium has a positive charge, yet the inside of the cell is negative. So sometimes I've seen students get confused on that. They're like, well, how can it be due to potassium when potassium is positive and the inside of the cell is negative? Well, it has to do because there's this big negative balance or deficit from the negative protein. So the potassium brings it up a little bit because those proteins are bringing it way down here. But the other thing is it ties to this pump. And having this constant change and imbalance means the membrane's always kind of chasing resting. So it always comes back to this steady state before the next wave of communication can be accomplished. But potassium is considered by far one of the more important ions in it, but it has to do with this pump because it's not an even 1-to-1. But we have some other pumps also that move potassium, as we'll come to learn throughout the semester. So here's what I was just drawing up. So the inside of the cell, it has a net negative. The outside extracellular environment has a net positive charge to it. So another way of saying that is that the inside has the excess of the negative, the anions, where the extracellular environment has the excess of the cation. So we can work out, there's actually a number, so we can work out the equilibrium potential for each ion in each compartment. So we have the concentration of potassium in the intracellular fluid is 150, and then extracellular is 5. And then look for sodium and chloride, they're way higher outside than they are inside. So this is the formula. I'm not going to ask you to use this formula. No way. But I do want you to understand what equilibrium potential is, and that each ion has its own equilibrium potential, but that does affect the overall electrochemical gradient. So if we want to figure out what the resting membrane potential of an individual cell is, we need to consider the concentration of all the ions at their equilibrium potential. So we're just looking at one ion in this example. All right, so this is what we actually do in lab, and we'll simulate it this semester since we're not in the classroom, but this is how we can plot changes in resting membrane potential. So the basic setup is we have a cell and it is in a saline bath, so to simulate the extracellular environment. There, the inside is negative, and one of our cell types has a resting membrane potential of -70 millivolts, which is why it's showing you that. So it doesn't actually mean the inside of the cell is measured at -70 all the time, because it's constantly changing because everything's fluid. What it means is that the difference between the extracellular and the intracellular environment is about 70 times different. And it's negative inside the cell, so that we get the negative symbol. So this setup is we have 2 voltage meters that are connected to the voltage meter to measure it, but then also to a graph. So 1 meter goes in the cell, 1 meter goes in the fluid. And then they go through and it's measured, and then we get a plot in the change of resting membrane potential over time. So x-axis is time in milliseconds. This is really quick. And then the y-axis is membrane potential, which is usually expressed in millivolts. So it begins at about -70, where it does, moving along, and then it goes down. This is called a hyperpolarization. Hyper means more or a lot. Polar means spread or opposite. So a hyperpolarization means we're going further down from 0 or further down from threshold. And then we repolarize and get back to resting, moving along. And now when we go up, that's called a depolarization. And the membrane potential increases and then it goes back down. That's another repolarization. So we use these terms when looking at electrical potential, when looking at studies of the nervous activity in the muscle. We also use them when we do electrocardiograms. So a hyperpolarization always means getting more negative, and then a depolarization means we're getting more positive, even though we're still negative below 0, at least in this example. Okay, so, oops, sorry. So putting it together, this figure really summarizes what I drew here. We've got our resting membrane potential of -70 millivolts. It is tied to the concentration of the ions and negative proteins, but which are moved back into place by, very importantly, by the sodium-potassium pump. Right? If it weren't for the action of the sodium-potassium pump, there would be no way to get back to resting membrane potential and no way to start over because there's no gradient. So the pump comes in when there's no gradient active, and it puts it puts it back where it's in higher concentration against the gradient. So there are some other differences that tie to why potassium is such an important ion in regulating membrane potential. It has to do to the fact that there are way more leak channels for potassium on our cells. And this is an example of a leak channel. So here's potassiums. So these are just channels that allow for the ion to diffuse through. So leak channels are going to allow for diffusion, but because there are so many more for potassium than there are compared to sodium, and then we get this uneven movement of potassium and sodium, our cells are about 40 times more permeable to potassium. And so that's another reason why potassium is really tied to driving that resting membrane potential. So the changes depend upon the concentration of ions, but that also is dependent upon the permeability on the membrane to that particular ion. I mean, if there's no channel for it to move through, it can't move, and then it can't change it, right? So the terms are depolarization, repolarization, and then a hyperpolarization. All right. that actually wraps us up for chapter 5. We will continue our discussion on resting membrane potential and the cell membrane as we move into chapter 6 and chapter 8, which is in unit 2. So next we're going to go to chapter 4, which is on metabolism, and that will wrap up the first unit. So you'll have your content for your first lecture exam. As always, feel free to email me. You're always welcome anytime, and I hope to see you in office hours. Take care. Bye-bye.